Hi, this is Dr. Martin, and this video will be about the Chapter 10 lecture, Part 2, General Characteristics of the Nervous System. A synapse is a site at which a neuron transmits a nerve impulse to another neuron. That means neurons communicate with each other at synapses. If a neuron sends impulses, it is located before the synapse. We will call this presynaptic neuron. If a synapse receives the impulses and located after the synapse, then it is called postsynaptic neuron. Between them, we have synaptic cleft, which separates the two neurons from each other. Synaptic transmission is one-way transfer of information. Impulse travels down the axon of presynaptic neuron to the axon terminal. When impulse reaches the synaptic nub, it causes influx of calcium ions. What that means? The impulse causes the calcium ions channels open at the axon terminal, and the calcium ion from outside of the neuron gets inside of the neuron. At the end of the neuron ending or axon ending, and when the calcium gets into the cell, when the calcium level getting higher into the axon terminal, then this leads the synaptic vesicles becomes part of the plasma membrane at the synapse and spill their content, which they carry the neurotransmitters via exocytosis into the synaptic cleft. Those neurotransmitters is not one type. There's different type of neurotransmitters. To be exact, there are over 50 different type of neurotransmitter that we will talk in next video. Those can be excitatory or inhibitory neurotransmitters. According to the type of the neurotransmitter, an excitatory or inhibitory effect on postsynaptic neuron will be expected. Let's focus the picture in this slide to understand the cell membrane potential. In this slide, you see the bi-layer phospholipid molecules resembling the cell membrane. And inside the cell, we call intracellular space, and outside the cell, we call extracellular space. You see the blue color coded side is extracellular, that means outside the space. And you see the ion distribution. Ion distribution is unequal. What that means, although all the ions can be found inside and outside, but it's not equally distributed. There is more sodium and chlor outside of the cell comparing to more potassium in the inside of the cell. Sodium is positively charged, and potassium, because potassium attached to the negatively charged amino acids and cause the inside of the cell being negatively electrical charged. And because of that reason, this unequal distribution of ions, a cell membrane is usually, during the resting, there is no neuron stimulation, electrically charged or polarized, so that the inside of the membrane is negatively charged with respect to the outside of the membrane. Important in conduction of impulses in neurons and muscle fibers to know the cell membrane potential. Let's summarize. Potassium ions are the major intracellular positive ions or cations. Sodium ions, major extracellular positive ions or cations. This distribution is largely created by the sodium-potassium pump, but also by the ion channels in the cell membrane. Sodium-potassium pump transports sodium ions out of the cell and potassium ions into the cell. Remember, sodium-potassium pump is a way to transfer or transport the ions in an active transport. It goes against the concentration gradient and it requires energy. However, ion channels formed by membrane proteins to help regulate passage of specific ions into or out of the cell according to the concentration gradient is passive transport. Many chemical and electrical factors affect opening and closing of gated ion channels. So every ion has their own gated ion channels. Sodium has, potassium has, chlor has. But sodium potassium pump is for two ions, sodium and potassium. Takes the sodium, three sodium ion to outside of the cell as an exchange of two potassium ions into the cell. The resting membrane potential, defined as the electrical potential difference across the plasma membrane when the cell is in at rest or non-excited state. And when the neuron is not stimulated, then electrical charge across the membrane we call resting membrane potential. Sodium and potassium ions follow the rules of diffusion. That means concentration gradient, right? From high concentration area, it goes to low concentration area. In resting membrane potential is considered as a minus or negative 70 millivolt potential difference, and that means the inside of the cell more negative comparing to outside of the cell. Membrane of a neuron is a polarized membrane with more potassium ions inside the cell than more positively charged sodium ions outside of the cell. And we have more negatively charged ions, proteins inside of the cell as well. So inside of the cell is negative relative to outside of the cell. And this amount is minus 70 millivolt due to unequal charge distribution of ions. If the resting potential changes, the sodium potassium pump will come and restore it. But remember, sodium potassium pump is an active transport and requires energy. Neurons are excitable cells. They detect the stimuli and respond by changing their resting potential. Common response is the opening of gated ion channel. If membrane potential becomes more negative, the membrane is hyperpolarized. If membrane potential becomes less negative or getting the positivity, closing the positivity, the membrane is depolarized. Remember, resting membrane potential was polarized and inside of the cell was minus 70 millivolt, more negative outside the cell. Now, you see the isoelectric line here in this graphic is minus 70 at the beginning in the left. So membrane potential, and when you look the time, milliseconds, in milliseconds, resting membrane potential is minus 70. But if the first thing is sodium channels, sodium ion gated channels opens when there is a, remember the neurotransmitter released to the synaptic cleft and attached to the receptors, the first thing happens, sodium channels opens. Sodium started getting into the cell, so cells starting to becoming more positive comparing to resting membrane potential. So polarized situation changes to depolarization. Local potential changes are graded. The greater the stimulus intensity, the greater the potential change. If degree of depolarization reaches the threshold potential, which is the limit to you can either have a response or not, this is the minus 55 millivolt, and then there is an action, we call this action potential happening. So threshold potential is minus 55 millivolt, and If the stimuli comes but cannot reach and exceed the threshold potential, there will be no response. So we call this all or non-response. If there is a potential reach and exceeded minus 55 millivolt, and you will have the exact same response. So all or none response comes from that. If degree of depolarization does not reach the threshold potential, the minus 55, an action potential will not occur, will not happen. In this slide, I just wanted to show you how the action potential pass through the axon length. You see the first thing happen, it's the sodium channels opening, and because sodium gets into it, into the axon, and it causes the positivity inside become positively charged. That's about all I wanted to show you. Now let's go back. At rest, we said the membrane is polarized, has a minus 70 millivolt electrical charge. And when the threshold stimulus reached the minus 55 millivolt, which is the threshold, and sodium channels open and membrane depolarize, it goes to the positivity toward the zero. And potassium then start leaving cytoplasm, and when the potassium starting leaving the cytoplasm, we call this the repolarization, because it's trying to be polarized again. However, potassium channels open slowly comparing to sodium channels. This slowness caused a brief period of hyperpolarization, and minus 90. You will see in the last graphic in the right bottom corner, and when you look, you will see the repolarization. After repolarization, it goes below, brief period below the isoelectric line. We call this hyperpolarization. Then sodium potassium pump takes over. It gives the three molecules sodium outside of the cell and takes the two molecule, allows the two molecule potassium inside the cell and restore the electrical charge. Axon hillock is the initial segment or trigger zone of the axon when the axon started to form from the cell body. And axon hillock has many voltage gated sodium channels, and during the resting membrane potential they remain closed. When the sodium channels, voltage gated sodium channels opens only when the threshold is reached, which is the minus 55 millivolt. And sodium ions diffuse into the cell, the membrane depolarize until it reaches the positive 30 millivolt. And in that moment, we say beginning the action potential. Sodium channels close in that moment, and potassium channels opens, and potassium diffuses out and membrane repolarize. Follow me on this graphic. As membrane potential then drops below the minus 70 millivolt, because of the potassium channels as kind of slow to open it, slow to close it. And the membrane is temporarily hyperpolarized, then potassium channels closes, the resting membrane potential re-established by the sodium potassium pump. Action potentials are propagated down the length of the axon as nerve impulses. Active transport re-establish the resting membrane potential of minus 70 millivolt as sodium and potassium concentration are restored. Concentration gradient are again maintained while the neuron is at rest. So an action potential is an all or non-response. If a neuron axon responds at all, it responds completely with an action potential, which propagates down the axon as a nerve impulse. A nerve impulse is conducted whenever a stimulus of threshold intensity or above is applied to an axon. All impulses carried on an axon are the same strength. However, stimulus of greater intensity may produce higher frequency of action potential. And that means impulses per second. You can have more impulses per second, but not stronger impulses. During an impulse, the portion of the axon actively conducting the action potential is not able to respond to another threshold stimulus of normal strength. This is called the refractory period and has two parts: absolute refractory period and relative refractory period. Absolute refractory period is the time when threshold stimulus cannot generate another action potential. All the voltage gated sodium channels are briefly not responding. Relative refractory period, however, time when only high intensity stimulus can generate another action potential. Repolarization is not complete yet, and membrane is re-establishing the resting potential. Refractory periods limits number of action potentials generated per second. The speed of impulse conducting varies with myelinization. Myelin, remember, is a rich in lipids, lipoprotein substance, and prevents water and water-soluble substances such as ions from crossing membrane, acts as electrical insulator. Ions can cross membrane only through the gaps in myelin sheath, we call the node of Ranvier. Myelinated axons transmit impulses through different conduction we will call saltatory conduction, and in which the action potential jump from one node to another node down the axon. And saltatory conduction is much faster than impulse conduction in unmyelinated axons. So axon diameter also affects conducting speed, and thick axons transmit faster than comparing the thin axons. Thick myelinated axons, the speed is in per second 120 meter. However, the thin and unmyelinated axons, the speed is per second is only half meter, 0.5 meter. And to understand the saltatory conduction, again, just see the node of the Ranvier between the, you see the Schumann cell represented here. Remember, Schumann cell found only in the perfect nervous system. The similar one that found in central nervous system we call oligodendrocyte. They all have spaces, and those spaces we will call the node of Ranvier. Transmission of a nerve impulse down a myelinated axon happens in this saltatory conduction way. So it's jumping, right? Jumping from one node of Ranvier to another. That's how the nerve impulse propagated throughout the concentration of potassium and sodium ion in the extracellular fluid and their permeability will affect the impulse conduction. If you have a higher potassium concentration in your plasma, and it will lead to excitable neurons, perhaps conversions. And in the same manner, if there is less potassium than normal in the plasma, neurons can become hyperpolarized. Action potential are not generated. The lack of impulses leads to muscle paralysis. And if there is a decrease in the permeability of the sodium ions, we use this reason for the anesthetic drugs. It stops the impulses from passing through tissue fluid around the axon. Impulses do not reach the brain, and there is no perception of the touch or pain. I hope you enjoyed and you learned. Have a good day.